Hello students, welcome to today's class. So today we are going to focus on aerobic respiration. We know that respiration is the process by which we are getting the energy, and when it takes place in presence of oxygen, then we are telling it as aerobic respiration, right? So we can see that the aerobic respiration is not simple, okay? That we have been studying in lower grades, the simple formula, right? That the glucose is broken down in the presence of oxygen and we are getting the water, carbon dioxide, and the energy, but rather we can see there are four steps involved. So the first step, we can see that the glucose was broken down and it was a partial like oxidation, and it was formation of three carbon compound. There were three carbon compound, that is glucose broken down into pyruvic acid. But now this is taking place, that glycolysis is taking place in the cytoplasm. In the last class, or if you check my last video, I will be linking it. It is there I have mentioned in details about the whole glycolysis process. So after the glycolysis is done, like we go from our school to our house or house to college, like that we go from one place to another with the help of taxi or the shuttle. So here also there is a kind of shuttle which is going to help the pyruvic acid enter the mitochondria, because the rest steps of the aerobic respiration occurs in the mitochondria, and that is why the shuttles are important, which is going to take place inside, like we help in like entering the pyruvic acid inside the mitochondrial matrix, for which there are two shuttles present: malate aspartate shuttle in the liver and the kidney and the heart, and glycerol 3 phosphate shuttle in the skeletal muscle and the brain. Okay, so like when we like book for a cab or let's say shuttle, we have to pay a price. So we are going to know in which shuttle we have to pay a price. In the next class, we are going to study about the balance sheet and also the electron transport chain we will be knowing. So once the pyruvic acid is entering, what it will do? It is going to further take place in the oxidation, because the pyruvic acid needs to be oxidized further, right? So it is going to take part in the Krebs cycle. But before that, there is a reaction which is linking our glycolysis and the Krebs cycle, which is also called as link reaction. And then finally, after the Krebs cycle, electron transport chain occurs. We will be knowing about electron transport chain in the next video or in the next class. Okay. Now, what is this link reaction? You could all see that it is also called as a transition reaction. So what transition is taking place? We can see here that the pyruvic acid, which was formed during the glycolysis, it is going to be forming acetyl CoA with the enzyme pyruvate dehydrogenase complex, which is basically formation of three enzymes. What are those? Pyruvate dehydrogenase E1, E2, it is dihydrolipoyl transacetylase, and dihydrolipoyl dehydrogenase E3. So here these three enzymes are taking part, and we will see oxidative decarboxylation. So what it is? Oxidation and decarboxylation. Decarboxylation means removal of the carbon dioxide, and yes, we can see that the carbon dioxide is being removed. Along with that, we will see dehydrogenation, because there will be reduction, right? So we will see oxidation, that of the remaining second part of the carbon is oxidized by NAD. So NAD plus is the oxidizing agent here, and it is going to be reduced to NADH, right? And along with that, when it is forming, we will see that acetic acid is formed, right, by gaining two hydrogen. Along with that, we will see that there is going to be addition of the coenzyme A. So the coenzyme A is added, that is how it is forming acetyl CoA. Okay, I hope all of you have written up till this. Okay, now as it is linking the glycolysis and the Krebs cycle, so Krebs cycle is basically citric acid cycle where there are 8 steps, and we can see it's a like a cyclic manner it is going to be continuing, because the product that has formed, that if you all could see, that is our oxaloacetic acid. it needs to be replenished, and we require acetyl CoA as the as our like entrant into the Krebs cycle. So you might have studied that respiratory substrates, there are various kinds, not only glucose. Glucose is the most preferred one, but proteins can also take part, even fats. But whatever respiratory substrate we use, like whatever we use, they need to form acetyl CoA by any process. Okay, we will be knowing in them. Okay, now the acetyl CoA and oxaloacetic acid, they are going to together condense, like condensation reaction takes place and forms the citrate, or we can say citric acid. Okay, so if you all remember that this Krebs cycle, or the like Krebs cycle or citric acid cycle, it is also called as TCA cycle. Why it is so? Because this TCA cycle is basically called as TCA cycle because of carboxylic acid. Which is this? It is our citric acid, because you can see there are three carboxylic acid groups present in the formula of our citric acid, and it is forming due to two carbon acetyl CoA and also due to oxaloacetic acid. Okay, so if you all want to write about it, you can take a note. Okay, very important. Please take a note about it. I hope now the screen is fully visible about the first step. Now, the second step, let us focus on our second step, class. So we can see here in the second step, there will be a dehydration. We shouldn't dehydrate ourselves. We should like hydrate ourselves. But here we can see citric acid. From here, there will be removal of H2O, means one H plus, and there is removal of OH as well. So one H is going to be removed from this second last carbon, and one OH from this carbon. And as it is taking place, we can see there is a double bond, and it is cis acotinate which is forming. Okay. So I hope all of you have written cis acotinate is forming with the help of the enzyme, that is acotinase. Now this much I hope it is clear. Now step 2B. Here we are going to hydrate ourselves. You can drink some water. Now we can see in the hydration the cis acotinate, it is going to form this isocitrate. Here you will notice that there is addition of one H plus and OH minus. So simple. You can all draw it with different color pen also, so that all of you remember that H and OH is added, right? So it is taking place with the help of acotinase, isocitrate is forming. So second step is divided into two steps. Now the third step is our oxidative decarboxylation. So what is forming here? Let us understand. So we can see that isocitrate, it is going to form an intermediate, that is oxalosuccinate, because first there will be dehydrogenation. That means with the help of isocitrate dehydrogenase, NAD is going to form NADH. So we will see 2H, that you can see A 2H which is present here, NAD. There was no H. Here we can see 2H. Where from it is coming? It is coming from the isocitrate. This 2H, they are removed, and it is formed oxalosuccinate, which is an intermediate. After that, there is going to be removal of carbon dioxide. So here, if you remembered in the citric acid we have studied, there were three carboxylic acid. Now you will all see that one carboxylic acid is removed, and as it is removed, so one carbon less, so it is 5 carbon. So it is forming alpha ketoglutarate acid, or in some textbook it is written alpha ketoglutarate, same thing. Okay, so first oxidative decarboxylation of TCA cycle in step 3. Step 4, same oxidative decarboxylation. We will see that there is going to be dehydrogenase, that is alpha ketoglutarate. Dehydrogen is going to work, and NAD will be taking the 2 hydrogen. And if you all notice very carefully that they are taking the hydrogen and they are forming the NADH and H plus, and along with that we will see there is going to be removal of the oxygen. So the last carboxylic acid is also now removed. So from 5 carbon we will see it is going to form 4 carbon succinyl CoA, and as the coenzyme A is also present in this reaction, so it is going to be also added. Okay, so now step 4 is clear. Let us focus on step 5. It is substrate level phosphorylation. where we see that the succinyl CoA, which was basically 4 carbon compound, it is going to be like helping in this process of phosphorylation. So that is why succinyl CoA synthase, it is going to form succinic acid, and we will see here that ATP or GTP is also released along with that CoA, that is coenzyme A is also released from here. It is forming succinic acid. Okay. Now the substrate level phosphorylation is clear. Now dehydrogenation. We know again there is going to be 2H which is going to be removed. As you can see here, 2H are removed here, and it is going to form FAD into FADH2. And we can see fumaric acid. Fumaric acid is forming, or we also tell it as fumarate. So that's why you can see here is a double bond as 2H has been removed, right? Now that we are clear about dehydrogenation, hydration, we need to hydrate ourselves. So the fumarate, here we can see, here there is going to be addition of H2O. So the fumarate is going to work here, and we can see one H and OH is now being added in this process. All of you please take down the note. So now we can see that the mallet that has been formed. So the last step, that is the step 8. So in the last step, we can see mallet, which is a 4 carbon compound. Now here also again dehydrogenation is going to take place, where 2H is going to be removed, and as it is removed, we can see it is going to form double bond with the oxygen. So it is COOH, it's carbon with double bond carbonyl, and we can see CH2COH is forming. So finally there are only 2 carboxylic group which are present now, and it is forming oxaloacetic acid, which is again going to replenish, right? So it is very important that oxaloacetic acid is forming, because replenishment of oxaloacetic acid is important for continuation of the TCA cycle. So if you all are wondering, it is very difficult. So now I hope it is going to be a little bit easier. First step, condensation of the acetyl CoA 4 carbon and the 2 carbon oxalo, sorry, my bad. So the 4 carbon oxaloacetic acid and the 2 carbon, that is acetyl CoA. After that, there is going to be dehydration, and in this dehydration, we are going to see how the citrate, from where we are going to remove the H2O. As the H2O is removed, we will see that the OH and the H here is removed, and it is forming double bond. And after the double bond formation, we will see that here there is going to be hydration again. So the cis acotinate which has formed here is now going to be added with H2O, and here we will see as the H2 is added, OH and H plus is going to be added here. Now that we can see it is forming isocitrate, let us focus now which step is going to take place. So we will notice that there is going to be oxidative decarboxylation. Here two things happens. There is going to be a dehydrogenation, along with that is basically oxidation, and along with that decarboxylation is also going to take place. So decarboxylation means that is CO2 is released. So we will see CO2 will be released, like if you see here, like this carboxylic group is removed as CO2. Along with that, we will see that here the H is also removed. This two H which are present here, these are also removed. That is why there is carbon with double bond O. And of course, NAD is released, which is our indirect ATP. First NAD is released. Again, if we come to the next step, we will see it is going to be oxidative decarboxylation. So the oxidative decarboxylation, here carbon dioxide is released. We will see that it is forming succinyl CoA. So from being 5 carbon, we will see now it is forming 4 carbon. First it was 6 carbon, right? So as it is removed, the alpha ketoglutarate, it is now succinyl CoA. The last carboxylic group which was here in the last position, it is removed, and we could see coenzyme A OH is being added as well, and there is also we will notice formation of NADH. Great. Now after this, we will see our fifth step is substrate level phosphorylation, where ATP or GTP is forming, and here succinyl CoA is going to form succinate, and we will see there is removal of the coenzyme A, CoAsh. Okay, in the form of CoAsh it is removed, and succinate or succinic acid is forming. Now succinic acid will be forming fumarate, or we also tell it as fumaric acid. But students, one thing is very important that all of you should note, that this succinate dehydrogenase, it is not present in the mitochondrial matrix, rather it is present in inner membrane of the mitochondria. Okay, please take a note about it, and we will see that in this dehydrogenation, the succinate dehydrogenase is going to take part, and we will see succinate is going to form fumarate. We will see the two H which were present here, they are going to remove, and a double bond is going to form. And after this, we are going to see that there is going to be hydration. Hydration is very important. Please keep yourself hydrated. So as we can see, there is double bond. Now the H2 is added. So H plus and OH will be added. So you can see OH plus is added in the second carbon, and third carbon H is added. So H plus and OH means hydrogen, this OH is OH group is added. Now after this, we will see dehydrogenation. So in the dehydrogenation, we will see this 2H, okay. This 2H, if I point it here, this 2H are going to be removed, and after removal, we will see that it is going to form oxaloacetic acid, and again due to malate dehydrogenase, here we will see another NADH. So 3 NADH are forming, 1 FADH, great, and we will see 1 ATP, and we can see 1 and 2 carbon dioxide, right? So what we can tell, what is our net gain? So let me remove the slides. Yes. So what we can find, what is going to be the net gain of our like whole process? So the net gain of TCA cycle. And how many TCA cycle? Only one TCA cycle. We could see there were 3 NADH, right? And we could see there was 1 FADH, okay? All these are indirect ATP, which is going to be oxidized in the electron transport chain, okay. And there was also 1 ATP. Along with that, we could see there was 2 carbon dioxide, right? But students, if you all remember, okay, what do we know? That there is like the pyruvic acid that was forming from one glucose. From one glucose, how many pyruvic acid were forming? If you remember, from one glucose, 2 pyruvic acid was forming, right? And from here we will see that the 2 pyruvic acid is going to form how many acetyl CoA? It is going to form 2 acetyl CoA. If it is going to form 2 acetyl CoA, then the products of the Krebs cycle is also going to be twice, right? So that is why we will see that Krebs cycle is going to take place twice, or the TCA cycle takes place twice with one glucose molecule. Okay. So I hope all of you have understood up till this. And now, if you are going to forget about the steps, okay, I have made it easier for all of you. I hope the slide is fully visible. Just a second. Okay. Now, I hope it is visible. Let me shift it here. So, it's a small rhyme to help all of you, know. So, let us summarize also. Acetyl-CoA, oxaloacetic acid by condensation forming citric acid, 6 carbon, right? So, after that, we could see that it is forming isocitrate, right? And that is was like hydration, dehydration. After that, there was an oxidative decarboxylation, for which there was removal of carbon dioxide, and 5 carbon alpha ketoglutarate was forming. Then we could see again decarboxylation, oxidative decarboxylation, succinyl CoA was forming. Then there was SLP, that is SLP means that is substrate level phosphorylation here. Due to susceptible phosphorylation, ATP or GTP was also released, and then we could see that fumaric acid was forming, then finally malic, and then by dehydrogenase we will see oxaloacetic dehydrogenation, oxaloacetic acid was also forming. So we could see that replenishment of this oxaloacetic acid is very important and crucial for TCA cycle. Okay, and now with the help of this rhyme, I hope all of you can remember citric acid can, isocitrate I, alpha ketoglutarate acid K, succinyl CoA S, and succinic acid some, fumaric acid M, that is funny, malic acid M, moving, and oxaloacetic acid only. So can I keep seeing some funny movie only? You all might be loving to see movies nowadays, right? So you can remember by this, can I keep seeing some funny movies only? Right? So can I keep seeing some funny movies only? That means can citric acid, I, isocitrate, keep, that is alpha ketoglutarate acid, and then we have seen, that is our succinyl CoA, and we have some, that is succinic acid, and we have funny, which is fumaric acid, and we have our movie, that is malic acid, and we have our only oxaloacetic acid. I hope now it is little bit easier. I hope so. Please practice all these steps, and I hope it is going to be easier, and all of you will master biochemistry as well. Okay, and this aerobic respiration and TCA cycling. So have a nice day, everyone. Take care. Thank you for attending today's class. Bye.